Welcome everyone. In this video, we're going to discuss chapter 36, which is blood type, transfusions, tissue and organ transplantation. Antigenicity causes immune reaction of blood. When a blood transfusion from one person to another were first attempted, there was a reaction. It can be immediate or delayed agglutination or hemolysis of red blood cells, which means breakdown of red blood cells, often occur, resulting in the typical transfusion reaction that were frequently led to death. Let's look at how we can stop this. Multiplicity of antigen in the blood cells, at least 30 commonly occurring antigens and hundreds of other rare antigens are present. Two particular types of antigen are much more likely than the other to cause blood transfusion reaction. They are known as OAB system of antigens and the RH system. OAB blood types include A and B antigens or agglutinogens. Two antigens, type A and type B, occurs on the surface of red blood cells in a large portion of human being. It is these antigens, also called agglutinogens, because of these often cause blood agglutination that cause most of the blood transfusion reaction. Major AOB blood types are: in transfusing blood from one person to another, the blood of the donor and recipient are normally classified into four major OAB blood types. It can be A, AB, B, or O. Genetic determinations of agglutinogens. The ABO blood group genetic locus has three alleles, which mean there are three different type of same gene. I here represents immunoglobulin and A represent the type of agglutinogen, which is IA mean immunoglobulin, agglutinogen is A, IB and IO. O allele is recessive to both A and B allele, which shows codominance. Let's look at the table. OO, OA, OB, and AA and BB and AB are all the alleles or genotypes found in the body. The blood type related to each allele are shown as: if you have genotype O, the blood type or the blood group you will have is O. If you have genotype OA or AA, since O is the recessive allele, it shows codominancy. You can either have blood group A or O. If you have allele OB or BB, the blood type you will have is blood group B. AB genotype will have blood group AB. Agglutinogens present in the each type of genotypes are: in AA, obviously agglutinogen will be A and the agglutinins will be anti-B. Because you have agglutinogen presence, then the agglutinin will be anti-B. For B, it will be anti-A. For A, B and O, there is no for O there is no agglutinogens because it is recessive, and for AB there is no agglutinin because AB blood group people can only donate blood to the AB group. Origin of agglutinins in the plasma. The agglutinins are gamma globulin, as are the most or almost all antibodies, and they are produced by the same bone marrow and lymphoid gland cells that produce antibodies to any other antigen. Most of them are IgM or IgG immunoglobulin molecules. The question arises is, why are these agglutinin produced in people who do not have respective agglutinogen in their red blood cells? The small amount of type A and B antigens enter the body in food, bacteria or other ways. These substances initiate the development of anti-A or anti-B agglutinin. The neonate has few if any agglutinin, showing that agglutinin formation occurs almost entirely after birth. When type A agglutinogen is present in a person RBCs, antibodies known as anti-A agglutinin will develop in the plasma, and for B, anti-B agglutinin will be present. Agglutination process in transfusion reaction. When the blood are mismatched, so the anti-A or anti-B plasma agglutinin are mixed with RBCs, they contain A or B anti-A agglutinogens respectively. RBCs agglutinin results in the agglutinin attaching themselves to the red blood cells. The binding causes the cells to clump, which is this process of binding and the clumping of cells is known as the agglutination. During ensuing hours to days, either physical distortion of the cell or attack by phagocytic white blood cells destroy the membrane of agglutinated cells, which release the hemoglobin into the plasma, which is called hemolysis of the red blood cells, thus destroying the red blood cells in the body. Acute hemolysis occur in some transfusion reaction. Sometimes immediate hemolysis of RBCs occur in circulating blood. In the case, antibodies cause lysis of red blood cells by activating the complement system, which release proteolytic enzyme. This is the lytic complex which will rupture the cell membrane. Immediate intravascular hemolysis is far less common than agglutination followed by the delayed hemolysis, because not only does there have to be high titer of antibodies for lysis to occur, also different type of antibodies seem to be required. Then we have the blood type. Figure 36.2 shows blood type showing agglutination of cell of different types of different blood types with anti-A or anti-B agglutinin in the sera. If the red blood cell type is O, there will be no anti-A or anti-B present. If the red blood cells type is A, because there is anti-A present, the blood type will be A. Because the anti-B is present, the blood group will be B. For AB, both antigen A and antigen B must be present in the blood. Table 36.2 lists the presence of positive or presence or absence of agglutination of the four types of blood. Type O RBCs have no agglutinogens and therefore do not react with either the anti-A or anti-B agglutinant. Type A blood has agglutinogens and therefore agglutinate with anti-A agglutinant. Type B blood has B and which agglutinates with anti-B agglutinin. Type AB has both, which mean they both have they have both A and B and agglutinates. Rh blood types. The plasma agglutinin responsible for causing transfusion reaction develops spontaneously, whereas in the Rh system, spontaneous agglutinins almost never occur. Instead, the person must be massively exposed to an Rh antigen, such as by the transfusion of blood containing the Rh antigen. The type D antigen is widely prevalent in the population and considerably more antigenic than the other Rh antigen. Anyone who has this type of antigen is said to be Rh positive, whereas a person who do not have this type D antigen, it is said to be Rh negative. However, it must be noted that even in Rh negative people, some of the other Rh antigen can still cause transfusion reaction, although the reaction are usually much milder. Let's look at the Rh immune response. Formation of anti-Rh agglutinins. When RBCs containing Rh factor are injected into a person whose blood does not contain Rh factor, that is into an Rh negative person, anti-Rh agglutinin developed slowly, reaching maximum concentration of agglutinins about 2 to 4 months later. With multiple exposure to Rh factor, an Rh negative person eventually becomes strongly sensitized to the Rh factor. A delayed transfusion reaction can also occur, although it is usually mild. Upon subsequent transfusion of Rh positive blood into the same person, who is now already immunized against the Rh vector, the transfusion reaction is greatly enhanced and can be immediate and as severe as a transfusion reaction caused by the mismatched type A or B blood. Erythroblastosis fetalis. Hemolytic disease of the newborn is known as erythroblastosis fetalis. This is the disease of fetus and a newborn child characterized by agglutination and phagocytosis of the fetus RBCs. In most instances of erythroblastosis fetalis, the mother is Rh negative and the father is Rh positive. Baby has inherited the Rh positive antigen from the father, and mother develops the anti-Rh agglutinins from exposure to the fetus of Rh antigen. In turn, the mother's agglutinin diffuses through the placenta into the fetus, and this will cause the RBC agglutination. Incidence of the disease: an Rh negative mother having her first Rh positive child usually does not develop sufficient anti-Rh agglutination to cause any harm. However, about 3% of the second Rh positive baby exhibit some significant signs of erythroblastosis fetalis, and about 10% of third babies exhibit the disease, and incidence rising progressively with subsequent pregnancies. Clinical exposure to erythroblastosis fetalis. The jaundiced erythroblastic newborn baby is usually anemic at birth, and the anti-Rh agglutinin from the mother usually circulate in the infant's blood for another 1 to 2 months after the birth, destroying more and more red blood cells. On that, one treatment of erythroblastosis fetalis is to replace the neonate's blood with Rh negative blood. About 400 milliliters of Rh negative blood are infused over a period of 1.5 or more hours, where neonate's own Rh positive blood is being removed. Prevention of erythroblastosis fetalis. Development of Rh immunoglobulin globin or an anti-D antibody that is administered to the expected mother starting at 28th to 30th week of gestation. The anti-D and antibody is also administered to Rh negative women who delivers the Rh positive babies to prevent sensitization of the mother to the D antigen. This step greatly reduces the risk of developing large amount of D antibodies during the second pregnancy. Administered anti-D antibody also attaches to the D antigen site on positive fetal RBCs that may cross the placenta and enter the circulation of the expectant mother, thereby interfering with the immune response to D antigen. Acute kidney failure after transfusion reaction. One of the most lethal effects of transfusion reaction is kidney failure. The kidney shutdown seemed to result from three causes. First, the antigen antibody reaction of the transfusion reaction releases toxic substance from the hemolyzed blood that cause powerful renal vasoconstriction. Second, loss of circulating RBCs in recipient along with production of toxic substance from hemolyzed cell and from the immune reaction often cause circulatory shock. The atrial blood pressure falls very low and renal blood flow and urine output decreases. The tubular hemoglobin's concentration to rise so high that the hemoglobin precipitate and block many of the kidney tubules, thus renal vasoconstriction, circulatory shock, and renal tubular blockage together cause the acute renal shutdown. Transplantation of tissues and organs. Autograft, isograft, allografts, and xenograft. A transplant of tissue or whole organ from one part of the some animal to another part is called autograft. From one identical twin to another identical twin, it is isograft. From one human being to another or from any animal to another animal of the same species is allograft. And from a non-human animal to a human being or from an animal of one species to another animal of another species is called the xenograft. Attempt to overcome immune reaction in transplanted tissue. Most important antigen for causing graft rejection are a complex called human leukocyte antigen or HLA antigen. Six of these antigens are present on the tissue cell membrane of each person. It is virtually impossible for two persons, except in the case of identical twin, to have the same six HLA antigen. HLA antigen occurs on the white blood cells as well as the tissue cells. Tissue typing for these antigen is done on the membrane of lymphocytes that have been separated from the person's blood. Prevention of graft rejection by suppressing the immune system. The T cells are mainly the portion of the immune system important for killing grafted cells. Their suppression is much more important than suppression of plasma antibodies. Some of the therapeutic agents that have been used for this purpose include the following. Number one, glucocorticoid hormones from adrenal cortex gland inhibits gene that code for the several cytokines, especially interleukin 2. These are the factors that is essential to induce the T cell proliferation and antibody formation. Number two is various drugs that have a toxic effect on the lymphoid system and therefore blocks the formation of antibodies in T cell. Number three is cyclosporin and tacrolimus, which inhibit formation of helper cell and therefore are specially infectious in blocking the T cell rejection reaction. And number four is the immunosuppressive antibody therapy, including specific anti-lymphocytes or IL-2 receptor antibody. This brings us to the end of the chapter number 35. Thank you for watching. If you like my video, don't forget to subscribe for more.